I'm going to give a little bit of scientific background, and I'll talk about the impact forensic science has had not only on DNA typing has had on forensic science, but also on other areas as well, particularly the criminal justice system in general. So we start here before DNA, okay? Early 80s, everything was calm and serene in our world of forensic science. We had dental records, we had fingerprints for personal identification. Forensic serologists, such as myself, we could identify some genetic markers in biological stains. ABO, we could do ABO blood type. We could look at, you could do RH typing of blood. You could look at polymorphic proteins in blood and other body fluids. Couldn't do very much discrimination of source, and so that it didn't have a tremendous impact usually, unless it was you got an exclusion. Microscopical hair comparisons, which is one of the things that I have done for many years, were routinely accepted by court. They really weren't open to challenge. And then we have the identification sciences, that's fingerprints, document examination, firearm and tool mark identification, just weren't challenged in court. They just waltzed right in and their results were accepted. I mean, there was no real serious challenge ever mounted to the scientific basis of these, whether a particular expert might be competent or not, might be raised, but these were not really subject to challenge. Within the forensic science laboratories, there wasn't very much standardization. There was an organization, ASTM Committee E30, that was formed in the 70s. Was coming up with some consensus forensic science standards, but wasn't terribly active and hadn't turned out very many standards. Very few labs did proficiency testing on a continuing basis. They tested proficiency when you were going through your training, and then never really retested, despite the fact that we know that people's skills often decline. Laboratory accreditation was wholly voluntary. This was the accreditation through the American Society of Crime Laboratory Directors. And there was no CSI effect. There weren't any television shows that really focused on forensic science and made people, have high expectations of forensic science. So everything was just really wonderful. Then some things happened. Well, first of all, DNA had been discovered about 30 years, the structure of DNA had been worked out about 25, 30 years earlier. And I bring this up because I wanted to make sure that you have an understanding here. If we're going to talk a little bit about the technology here, you have a double-stranded DNA of phosphate sugar backbones. These are sugar molecules. Those are phosphates. And you have these different bases, this adenine, thymine, guanine, cytosine. And these match up, these pair up. And even when Watson and Crick published the structure originally, they said, we can think of a way that this stuff can be replicated. It's basically, if you strip the strands apart, you provide the bases, they'll match up like they're supposed to. By the way, you can separate this duplex DNA by heating about between 90 and 95 degrees Celsius. You'll pop the strands apart. That's an important part of some of the techniques we use. Nobody really anticipated we could go back and we could look at old DNA. We could go back 10,000, and now we're back, tens of thousands of years with, being able to, extract, valuable information out of DNA. In 1983, a guy named Alec Jeffries, that's the guy right there, when he received an award at the FBI, I thought, Hoover's got to be drilling a hole in the crust of the earth. I mean, he's got to be spinning his grave like crazy, because this guy looks like a hippie. He's wearing sandals and he's bearded. The FBI's idea of, their agents going undercover, they loosen their ties. Let's say they're much more casual than they were back then. Anyway, he was looking at the human myoglobin gene, and he discovered what are called variable number tandem repeats, or VNTRs. He just found that he was cutting out the gene, and he was centrifuging it and collecting that strand of DNA, and he found that when he cut it out, that sometimes he would see other little adjacent bands in the ultracentrifuge, and he realized that there were strands that had different masses, different lengths that he was looking at. And he then went on to develop special DNA probes, sections of DNA that were labeled originally, radioactively labeled, that would actually bind to specific areas. And he actually was the first person to use this in any kind of a forensic investigation. It was actually a question of parentage, whether a person who was being presented, as the child of a British national was in fact really that person's child. About the same time, Kary Mullis developed another technology which would ultimately grow and take over DNA profiling, what's called the polymerase chain reaction, or PCR. And we're going to look at these. Before you look at the different methods here of profiling DNA, I need to say something about how you get the DNA. People still use organic extractions, basically use use enzymes to degrade the proteins in your sample, detergent to disperse the cell membranes, and phenylchloroform to extract all this debris. And then you can precipitate the DNA strands. It's kind of fun. It kind of looks like little white threads in the cold ethanol solution. A researcher named Gill came up with what's called the differential lysis method, where he found that you could get the DNA out of the epithelial cells. You have a body fluid, like a semen stain. You're going to have epithelial cells from the victim, probably some urethral epithelial cells from the perpetrator. Then you're going to have all these sperm. What he found that by using this technique, he could get the DNA from the epithelial cell fraction and then go back and then also get the DNA from the sperm. So you could actually separate the fraction predominantly from the victim, and then you could get the perpetrator's DNA separately. Now, we were never able to do that when we looked at genetic markers, ABO blood group substances in dried semen stains. We could never tell who contributed what. And so there was always the possibility of the victim's genetic profile masking that of the perpetrator. So it really degraded the value of the evidence. And this was a big advance. If they hadn't done anything else, this would have been just really wonderful. You can also, if you have something that you think is just blood and not just a blood mixed with something else, you can just boil it up with polymer beads. You can take the stain, cut it up, and just boil it, and you will get the DNA out. Once you have the DNA, you could do the VNTR method. That came online first. And VNTRs are just segments of DNA that you cut out. This is a strand here in a certain area of your the DNA on one of your chromosomes, and up here you have here's the repeat pattern, and you might have 12 of those patterns repeating down here. You might have 17 here, and if you cut these out of the DNA with a restriction enzyme that could cut ahead of and behind this, you would get different lengths of DNA, and then you would just do electrophoresis, gel electrophoresis, run it through a gel that would act kind of like a sieve, and the smaller fragments would move faster, and you could separate out the different size fragments fairly easily. And the process was a little kind of involved. You do the restriction enzymes, you do electrophoresis. This is with using a fluorescent marker. You'd have to transfer the DNA to a porous membrane, nylon or nitrocellulose. Then you would take this membrane, you would then soak it with one of Alec Jeffrey's probes. The probes would bind to select the specific areas in the DNA, and then you take that and you would just slap a piece of X-ray film up against it and take a picture. And that generated these barcode type images that you see over here. Okay, this is actually from a paternity test, and this is one of the areas that we use for where this DNA profiling is used pretty routinely now. And even in criminal cases, sometimes we use paternity testing approaches. We might want to use reconstruct of one case, for example. They had to reconstruct the genetic profile of a murder victim, because the murder victim's body was never found. but she had a child, and we took, you could take the husband and the father and the sons, and you could then, by elimination, you could work out part of the mother's genetic profile. You would then treat this with, you could treat the membrane, you could strip off the probes for one set of markers, put another set of probes on, and you generate like 5 or 6 of these so-called autoradiographs. Okay. Cumbersome, time-consuming. This is why you couldn't get the results in a day or an hour, because it would take you about a week to get all of the autoradiographs, just after you've done the gel electrophoresis and everything like that. So it's and it required a lot of DNA. A semen stain about the size of a quarter. That'd be about what you could work with. And then the blood was about 50-cent piece, something about on that order roughly. And they pushed it down, but still, you required a lot of a fairly large amount of sample. Okay. With having this technology available, the FBI formed the Technical Working Group for DNA Analysis Methods. It was called TWIGDAM. And actually it's now called SWIGDAM. They decided Technical Working Group is a little bit hoi polloi. We should call ourselves the Scientific Working Group. But it's still used, still in existence, and still very useful. It came up with, let's come up with some methodology here, some standardization. And they also, saw the perspective here. If we can get, information is very easy to store. We have the sizes of the fragments. We can work that out. We can store that information in a database, and then we can query that if we have, DNA from a case where we don't have a suspect, and we can see if that person is already in our database. Congress passed the DNA Identification Act in 1994. They provided $40 million over like 4 years, and it was to establish the combined DNA index system, or CODIS. And this has been one of the most powerful tools available for criminal investigation. And I'll give you some examples of how this has been useful later. They also established the DNA Advisory Board, which was to come up with basically ways of, standardize a way of doing these kinds of testing, and it was a kind of a carrot situation. The money was offered to the state, realizing that the FBI, the federal government, has very limited criminal jurisdiction. Most of the crimes that are committed in this country are not federal crimes. Okay, where rape's not a federal crime unless it occurs on a federal installation. So you need to have, if you're going to have a database, you need to have the states participating. So the money is to get the states to participate. And so the terms of the law were, okay, you could get the money. You had to put up, the states had to put up a certain percentage of the money, almost half. They had to put it, but that was usually in terms of the salaries of the analysts. That would count. The FBI, the federal government would put up other stuff. There are a couple of provisos. They were not eligible if their state didn't have a DNA database law. So this was not very subtle suggestion. You might want to have a DNA database law. So, for example, I was on the panels determining who was getting the money all 4 years of this. And the first year we had a submission from the state of Vermont. A very nice submission. We might have suggested it be funded, but Vermont had no database law. So we said, sorry, you can't have it. It's a nice proposal. We probably would have given you some money, but you can't get it because you don't provide, your state hasn't established a database law. So guess what? Vermont promptly passed a database law. They could read the handwriting on the wall. It was a good idea to do it. And then they could become eligible. So that was the encouragement. They also had to comply with the methodologies of the FBI, which specific VNTRs you looked at, how you made the size measurements, and a bunch of other things you had to comply with to get the money. Okay. But this allowed them to get a whole bunch of states involved in doing this, putting everything, putting the data into the system. By about 1998, they basically became fully operational. Virginia was one of the states that got involved very early, and so at one point, 45% of all the samples in the database were from the state of Virginia. Now, what of course states often did is, well, we have a database law, and usually they started off, what they started off with was people who were convicted sexual offenders. And then they went to violent offenders, and then they just went to convicted felons. And now they're starting to, collect DNA samples from people who are just being arrested. And so now that's getting to some interesting privacy issues here. But the idea was, at least why focus on sexual offenders? Well, rapists are usually, they're usually repeat offenders. So it makes sense to have a database of those people. And so, we had a case, one of the first cases in Virginia where they used this. They had a guy who was, he'd been 2 months out. He went and committed a rape. And guess what? They did a DNA profile. He's in the system. He's arrested. He's, he's back in prison very quickly. And usually these people would go, they might commit a dozen rapes before they got caught again. So there were a lot of women who were spared being his victim, okay? So this became, now available, is very extensive, has now like millions of samples in it. So it's become extremely important, useful. Now, obviously, the defense bar didn't just let this in. They said, well, it's novel science. At least it can't be admitted, just walk in the door. They have to go some hearings. And there were a whole series of Fry hearings. This was pre-Daubert initially. And I actually testified in several of these. One in the district where the judge wouldn't let it in Minnesota, where the judge would let the results in but wouldn't let any of the statistical discussion in. That was the case where the Supreme Court said you can't talk about the statistics. That'll addle the jurors' minds. Okay, they complained there was a lack of standards, and initially that was a good point. You had private labs doing this initially, and there was no lack of standardization among the labs. And then people began to complain about the statistics. First, the databases were too small. Well, we've gotten past that. We didn't know what the error rate is. What's is it the error rate of the technique itself? Is the error rate of the labs? There was to and fro and arguing about that a lot. Another was an argument about when you're calculating the probabilities of a random match, whether you could use the product rule, where you would take the results from one locus and multiply those by the probabilities for another locus, because that gives you really, really tiny probabilities of random matches. And people argued, well, that there's population substructure, so that it invalidates this rule. Some of the populations aren't, as they say, in Hardy-Weinberg equilibrium and so on. And whether the databases were often set up according to race, which is kind of to some degree a social construct, and people basically self-identify what race they belong to, perhaps not correctly, in terms of their actual biological ancestry. And they objected to that. And there were some cases where a particular racial database was used, like somebody used a Hispanic database in one case, and the FBI expert testified that they'd used that one. And they talked about they did a general Caucasian database and then a Hispanic database, and ultimately it was overturned. The conviction was overturned. Said, look, it's very prejudicial. The guy is Hispanic. He's obviously Hispanic, and so you focused on the perpetrator had to be Hispanic. So you really tilted you could have really biased the jury. Obviously, these did not in the end prevail because DNA profiling is accepted. There were privacy issues. These come up. We're giving these people the DNA and they can find out all sorts about our genetic diseases and so forth. And in general, until very recently, we've been terribly interested in that because the genes about disease aren't terribly informative from an identification standpoint. The functional genes aren't terribly interesting. We look mainly at areas that are not part of functional genes. Why? Because they're not constrained as much. They undergo mutations much more rapidly, so they accumulate a greater variability. So they're much more useful from an identification standpoint. Okay, let's back up now to Kary Mullis, okay? That's Mullis. He got the Nobel Prize, by the way, for this. He was working for the CODIS Corporation and tasked with coming up with a way of making strands of DNA. And he got the idea, let's hijack let's just hijack the system that cells use. Let's take our DNA. We have a DNA target sequence right here, okay? And what we're going to do is we're going to provide a little piece of DNA that will just match a part of the genetic code right here. There's another primer. These are called primers, another little segment right here. We're going to split the duplex DNA apart by heating. Go up to above 90 degrees Celsius. We'll strip it apart. Then we'll cool it back down. The strands aren't going to immediately go back together. And so we're going to put in these little primers that can bind in areas. These will have a genetic code that will target specific areas. So we can, it's kind of like this. I like to say PCR is like having a Xerox machine. It's not like we're copying pages from a book so much as they're sentences or paragraphs on a particular page. So we can specify which page we're going to look at and which specific paragraph we're going to make copies of, because the segments that we're typically making, these pieces of DNA that we're making, go up to, as you'll see, go up to about 400 base pairs. So they're not really big. Anyway, so the idea is you also provide a soup of bases that need to go in there to make more strands of DNA. You also provide an enzyme called a DNA polymerase that will stitch these together. So you just make a new DNA strand right here, make a new DNA strand here. And then the idea was, well, okay, it's really clever. We'll just reheat it. Okay, this is done. We go to high temperature, elevated temperatures to split this apart. Then this binding takes place. We lower the temperature, the binding takes place, the extension takes place. Then we heat it up again and we split the copies off. And so we're making copies of copies. So we would do, effectively, we're almost doubling the amount of that particular segment of DNA we've got each time. So we could go 25 cycles or something. So that's about 2 to the 25th power number of the copies. The actual original DNA is actually lost in there. We probably don't really even see it. Yeah.
The primer attaches to where it is because you set it up to be like match certain.
It matches a certain sequence. Yeah. Yeah, exactly. It has the complementary sequence of bases, and it's a short piece of DNA. Now we can do all sorts of things to that. One of the things you can do with it is you can stick a label on it. And what's normally been done is to put a fluorescent label, a molecule that actually fluoresces and fluoresces at specific colors. So you can actually tell which area, when you separate the DNA strands, you can tell where they're coming from. There are a whole bunch of tricks here, really, that you can do with this, as you'll see. There are lots of fun things you can do. And you can do and the nice thing about it is you can be multiplying, you can be making copies of different areas simultaneously. Originally, people were just doing one locus. I think the original kit was doing the antigens that affect tissue transplantation, HLA-DQ alpha, human leukocyte antigens. And that was actually, a kit was developed initially for the clinical community, but had nice variability, so it was used, the first one actually used, first PCR kit used in forensics. And it was it's a great technology. It's quick and dirty. You're talking here when you're doing 25 cycles, you're talking maybe an hour to do the amplification. The problem, of course, is you get lots and lots of DNA, so you have potential contamination problems that are rather significant. So,
excuse me, does the copying process introduce errors?
It can, but these are pretty high fidelity, and they've been very careful about making sure that they select areas where they get high fidelity. There are some types of sequences that give problems, and they stay away from those. They don't use those for the kind of loci that we look at. Also, we very careful about it, make sure the conditions are good for high stringency binding. You can have low stringency where you can get mismatches. And so we use high stringency conditions so that you reduce the number of mismatches. People very carefully, that's an important question, and that was something that was very carefully looked at when they decided to do this. A lot of this was done commercially. PCR testing is focused on what are called short tandem repeats. These are areas, they're like VNTRs except they're a lot shorter. They're usually 4 base pairs that repeat. Okay, this has 7, this has 6, and you just make these little segments here. They're different lengths, and so what you do then is you separate them by electrophoresis. In this case, they use gel electrophoresis, and here what they have is, you're seeing up here is the different loci. They're different lengths. They go up to about, it's 100 to about 2 something up there. What is it? It's up to about 320. Okay, and these are the numbers label the different. They've just been identified the number of repeats, and so this individual has in this particular locus has 15 repeats here. They're homozygous there. They're heterozygous at the other 2, and so on. So that you can. Here they have, that's 14, 16, these are the number of repeats. And so they've looked here, they just amplified these 3 loci, and they did electrophoresis, and you get this nice pattern. Well, since you can do number of loci simultaneously, you have real capability here of really cutting, really industrializing the process, gaining economies of time. So, the FBI came up with, here are the 13 loci we're going to use in our CODIS. These are the core loci. You can use other stuff if you want, but you want to participate in CODIS, you're going to use these 13 STR loci. They're all designated, and you can see they're indicated which chromosomes that they're on, which chromosome pair they're on. The amelogenin ones down here, right here, that's not actually an STR. Those are not STRs. Those are actually code a protein or an enzyme that's involved in the production of tooth enamel, and they're used because the segments that you're making copies of are different lengths on the 2 chromosomes. So it's a really good sex marker. If you see two peaks at that in the gel electrophoresis where the amelogenin fragments come out, okay. It's got to be male. See 1 female. So it's kind of cool. You can get that immediately, out of your data, and you incorporate this into your kit. Okay. So actually, here are the kits they're now using. Okay. The one on the left there, this Applied Bioscience produces this. Okay, and these represent the colors of the labels, the fluorescent labels. Okay, and so these are where they show up. So you can have them overlap. Find where the peaks would be in the same general area, but because they're labeled differently, and you scan your products with a laser, you can tell which locus they belong to based on where they are in the color. And Promega has this one over here, the Promega 16. It has a whole bunch of these, and it's kind of cute. It's really kind of a neat idea. We can really industrialize this process. We do 1 amplification, and we get out all of this information. We just run it then through. We do slab electrophoresis, or we do capillary electrophoresis. When we started doing databases, they began to think about, well, maybe we ought to have capillary electrophoresis systems that can run large numbers of samples, because you have to do a database. If you're doing databasing, you might have 10,000, 20,000 samples you have to process. Bodie Technology, which is down in Southern Fairfax, they've been a contractor for doing a lot of this work, and they basically have these FM BIOS systems that they just would run basically almost 24 hours a day, basically till they burned up or something. They would come in, they'd set up their gels, then run them in the afternoon, and then read them, and they would just, they had shift work, so they had shifts coming in, and they were just running these things. So they had to do they took a contract from South Carolina, 17,000 of these things. Now, what's the impact of all this been? Well, first of all, I've kind of already alluded to this. First of all, the concept of cold hits. You don't have a suspect. You have an unknown subject sexual assault case. You don't know who did it, okay? You don't have any idea, okay? If he's in the database, you can find him. So you do the DNA profiling, you send it to CODIS, and maybe you get a hit. And frequently you do. I mean, we're probably at the point where we're probably getting several, most states are getting a couple of hits a day, not a day, but every week, probably on that kind of order. And actually, you can, and then you have these are the number of cases that are helped by these inquiries. This is actually a statistic from a publication of the FBI, as the number plotting the number of offender profiles that you have versus the number of cases helped, and you see that just nice linear relationship there. The more offender profiles you have, the more cases that you help solve. The other kind of cases you can look at are cold cases, cases that occurred before DNA profiling was even available. If the evidence has been preserved, you can go back. And if it's been properly preserved, you can go back, you can get a DNA profile off of it, and then you may be able to convict somebody that was already a suspect but you didn't have a real strong case against, or somebody you didn't even suspect. We'll talk about a cold case, a particular cold case in a minute. You got these, sometimes people are now finding you can occasionally run a DNA profile to get into CODIS. You don't get an exact match, but you find something that's close. Maybe that's a family member. California's been using familial DNA for a couple of years. It doesn't match Joe, but it might be his brother John that did it. So you start investigating him, look at his background. How old is he? Was he old enough to have committed the crime? What do we know about where he was at when the crime took place? Now, Maryland recently, of course, repudiated the use of that.
Is that just used as an investigative tool?
Yeah, basically you don't use it for conviction. You've got to do an investigation. This guy might have a couple of siblings. You don't know which sibling it is. Find somebody, is there anybody, or maybe none of them are possible. Okay, then you kind of have to drop it. If you have somebody that is a viable candidate age-wise, okay, can you get some other information that suggests he might be it? He might be the person. Then you have to go get a judge's warrant.
They found him because of his brother. He was like a sleepaway, some kind of sleeping killer or something, or he used to kill people. And then they got his DNA. So they found the identity.
Yeah, they've done that. Yeah, they did. I think it was one of the California cases. They don't do a lot of that. I mean, it really doesn't come up that much. I mean, right now we don't have that many where you get occasions where you get get close but not exactly a match. And then finally, people who claim that they've been wrongly convicted, maybe there's DNA evidence that will exonerate them, so they could go back and look at the DNA evidence and have it redone. That's been a big push of the Innocence Project. I've worked with Barry Scheck and Peter Neufeld on a lot of cases over the last few years, and they've been going back and they've been doing a lot of post-conviction DNA testing. Sometimes it's useful, sometimes it turns out not. Okay, so let's take a local cold case. I decided to talk about some actual cases. Instead of showing you diagrams of molecules, let's actually look at some people here. So we'll go back to May 1988. Veronica Tina Jefferson, she's the young lady up there at the top. She was raped and fatally shot in Arlington. In December ’88, Rachel Raver and Warren Fulton, who were George Washington University students, were abducted as they left after they left a restaurant in D.C. Were taken to Reston. They were both shot. Miss Raver was raped while she lay dying. And they had no suspects. These cases they had the evidence. They'd done all the investigation they could, but they had no witnesses. They had no viable suspects. Now, some of our student interns got involved in this case. They were student interns in Fairfax County. They had the Raver and Fulton case, and they suggested, well, we have the technology now to DNA profiling. Let's have that evidence. Have the DNA profile on particularly the vaginal swab. Let's have that done and see if we can find get a hit in CODIS. So what they did is first thing happened in year 2000, they did the DNA testing, and they did the DNA testing in the Jefferson case, and they linked them. The same person committed both crimes, okay? They submitted to CODIS, but nothing came back. 2005, they decided to submit again because there may have been more samples added, more offender samples added. Now they got hits to Alfred Preto's DNA. Why was Preto in there? He had been convicted of a rape murder in California, and that's why his DNA was finally put into the CODIS system. He was on death row in California. Now, if you know about California, he would probably die of old age or slip and fall in the prison shower before he would be executed, okay? In fact, I was discussing this case with one of my colleagues who had been involved in a case in California in mid-'80s, and he said, the guy was convicted, condemned to death, he's still alive. Okay? The mills of the wheels of justice turn very slowly there. Any case, he was extradited to Virginia, tried for these murders, condemned to death. There was some toing and froing. The first trial was thrown out because one juror claimed that have been coerced into convicting him. Then he was convicted and condemned to death again, and then there was an issue about the jury instruction, whether that was right, and it got overturned, and they just had a hearing on the death penalty. He was condemned to death again. So he's now on death row. I would say that his life has been, expectancies have been materially shortened at this point, because Virginia will get to him very quickly. However, in the meantime, he's been also linked to 2 unsolved murders in Riverside, California. He committed 2 other rape murders there. He was a busy little soul, okay? You could see where it would have been nice if we could have had the CODIS available much earlier. We might have got him before he committed some of these other crimes. He might have known, been able to nail him. But we may have other ones as well. There's some talk that he may enter into some sort of a plea agreement if he provides other information, either, I don't know about the Riverside cases or perhaps about some others, because this guy's probably been busy, committed a number of these. So there are a number of unsolved cases. There are at least some he had some surviving victims that we know, because some of them testified against him in California. Okay, post-conviction DNA testing. Probably the poster boy is Clerk Bloodsworth. In 1985, he was convicted of the rape and murder of a 9-year-old girl in Rosedale, Maryland. He was condemned to death, got a new trial, was condemned to death again. I was a defense consultant in this case, not on the DNA issues, but on the issues of some trace evidence that was found on the victim's body. There were some carpet fibers that were consistent with automobile carpeting. And the lawyer who was handling the appeal thought we might be able to introduce some new evidence, and he got a sample of he had a suspect he thought that had committed the crime, and got a sample, found the guy's car years later, found the car, got a sample of the carpeting. We looked at it, unfortunately, it wasn't even close. And so we didn't have anything to go forward with. It was interesting that we found the slides with the carpeting at the courthouse. They weren't in the forensic laboratory. And that's where the victim's underwear happened to be. We didn't know it at the time. And in '93, the DNA testing of the semen on the victim's underwear excluded him. He didn't provide the semen. And 10 years later, the DNA testing identified the actual murderer as Kimberly Shea Ruffner, who had been convicted of a crime shortly after Bloodsworth and was in the same prison, was in fact one tier below him in prison all this time. And they met and so forth. He didn't know that Kurt did not know that he was doing Ruffner's time on that case. In any case, in 2000, the Innocence Project, the Innocence Protection Act, which from law passed by Congress, established a post-conviction DNA testing program so that there'd be some funding to go forward and test some of these claims. By the way, that's he's showing himself what way he looked at the time that he was tried. So he's kind of the poster boy for this. I mean, he came close to being executed. There was a good bit of, examining the case overall, there was a good bit of prosecutorial and police misconduct. That's how they got some of the convictions overturned. There were some bizarre statements about shoe impressions that found near the victim, supposedly the same size as his shoes. And they were completely off. I mean, it was just, they didn't even have expert testimony. They had some police officer.
When did he get out?
He was released after sometime in around 1993, because he was excluded. So then they had kind of, hold on to the guy, well, we think we'll just keep him anyway.
Well, they sometimes do that.
Well, sometimes it takes a while. Sometimes it may be arguable that the evidence, that finding somebody else's DNA there doesn't necessarily exonerate him. In this particular case, it clearly did because it was the semen from the perpetrator. But in some cases, it's a little bit more ambiguous. And sometimes the guy may actually be guilty of something else. All right, what about, how did the DNA impact forensic laboratories? Well, cause standardization. We had SWIG-DAM, or TWIG-DAM, then SWIG-DAM, the DNA Advisory Board. But it had impact, as we'll see. It resulted in a lot of standardization in other forensic disciplines. Part of the standardization in DNA came from the fact that everybody just buys, they buy kits either from Applied Biosystems or from Promega, one of the two. Everybody's using the same kits, the same instrumentation, so pretty much everything is done exactly the same way. Documentation. Well, DNA typing, DNA profiling generates a lot of documents, a lot of computer files, so there's a lot of stuff that can be reviewed by a defense expert. There's a lot of stuff that you can have a technical peer review very easily, have another expert go over it and say whether would reach the same conclusion. Proficiency testing became very important because the defense bar constantly asked about, well, have what are the results of your proficiency testing? And that is now built into the accreditation process under the American Society of Crime Laboratory Directors Laboratory Accreditation Board. Cleanliness. We got real concern now about cleanliness not only at crime scenes but in the lab, but also at crime scenes, because we can't have contamination. We're amplifying stuff, and so we have to be careful about make sure we've wiped down all the work surfaces. In the crime scene, we've got to make sure that we glove up and we constantly are changing gloves so we don't transfer DNA between evidence. We're gloving up so we're not leaving DNA evidence behind. Then that's kind of a problem with some of these cold cases because if they're old enough, nobody took any particular precautions. And so there may be DNA there from an investigator. I can think of one case where it may have my DNA in it because I collected some of the evidence, but you weren't taking strict precautions. In some cases, there was accreditation was mandated. Yeah.
So when you mentioned the two labs, have they done lab reliability? In other words, are they 100%? You said the sample.
Pretty good, pretty close. Yeah, and NIST is involved in a lot of that testing. With that, they produce standard reference DNA samples for testing. Yeah, so they've been looking at that for a long time. Now they found with RFLPs that there were often discrepancies in the sizes that people were reporting for the same samples, but with PCR, it's pretty much cut and dried. One of the other things there, that gentleman there is Fred Zane. Fred Zane was a West Virginia, worked in the West Virginia lab. He was a state trooper. He worked there. He was a serologist. He was there for about 8 years. He went to Bayard County in Texas. That's San Antonio. DNA exposed the fact that he was basically dry labbing most of his work. He had a case where he said that somebody was included as a possible source of biological material. DNA testing later showed that he wasn't. Somebody had your hand up over here? Yeah.
Yeah, I know what dry labbing is only because of a recent.
Dry labbing means basically you don't actually do anything in the lab, you just make the results up.
Yeah.
And that's, he was doing a lot of that. He just wouldn't run samples. There's a recent case, if you follow the news, of Annie Dukin in Boston. She's actually been arrested and charged with, her cases at least 1,000 cases, and where their people were convicted. In Zane's case, the State Court of Criminal Appeal in West Virginia, when it came out that first that there was a DNA discrepancy, and then people started looking at his work and then realizing, hey, this guy's just faking it. He's been faking it for years. The whole thing was investigated by the State Court of Criminal Appeals in West Virginia, and then they said the State Police Crime Laboratory would have to become accredited by ASCLD lab. They wouldn't have a choice. They would be required to do that. They don't have a choice because of this. He caused problems down in Texas. There's one of the he led to a lawsuit against the medical examiner's office that runs the crime laboratory. by a guy who might very well have been guilty of a particularly atrocious rape, murder, but because of Zane's misconduct and other things, he wound up, the guy got it released from prison, turned around and he sued.
Did Zane get jail?
Nope. He was actually acquitted on one occasion. He died of cancer. Yeah.
Did they ever give any like motivation? Were they just overwhelmed? Like what was?
I think in his case, I met the guy a couple of times and actually looked at some of his casework. I never thought to actually look for the, I just looking at the report, reevaluating the reports. That taught me a lesson. I want to see everything. I want to see all the documentation, all the lab notes. I think it was partly maybe overwhelmed. I know when Oscar Davis sued Bayard County, his lawyers asked me to look at what case notes that they could find of his, and I ran through about 25 or 30 cases. These were mostly rape cases, and he was dry labbing a lot of them as no evidence of sexual activity. And I was fairly convinced he wasn't doing any testing. It was just very easy to write down no evidence of sexual activity. And there was unknown subject rape cases. They're not going anywhere. This is pre-CODIS. He knows they're not going anywhere, so he takes them. They're off the books now. They're not open cases anymore. They're closed, as far as the lab's concerned.
I mean, what kind of what would his caseload be? Like, what were the kind of numbers of?
I don't know about that. I mean, in Bayard County, he was the only serologist, so it might have been fairly large caseload. But part of it was just to ingratiate himself. He was viewed with great favor by prosecutors in West Virginia. I mean, so he was, he got convictions, and so he was really, very beneficial to them. So he ingratiated himself. I feel, from meeting him, the guy came across as pleasant and accommodating, and I thought was, in retrospect, well, the really bad people don't look as sinister as you'd like them to. So appearances are deceiving in such cases. Okay, well, let's go ahead. Mentioned the standardization, and we mentioned technical working group for DNA analysis methods. Well, they've just proliferated. They've sprung up like toadstools after a rainstorm. Here's just a partial list. I mean, a very partial list. In fact, we've got a notification from the American Academy of Forensic Sciences that they have documentation for the establishment of a scientific working group for medical legal death investigation. Okay, that's going to be a new SWIG. Okay, and you've got SWIG that's forensic anthropology. You've got SWIG drug, SWIG dam, of course, the old one. You've got SWIG GSR, gunshot residue. The only one that's still a technical working group is TWIGFEX, that's fire and explosives. We don't care. We don't need the extra prestige. There's SWIGIT. There's imaging technologies, illicit business records. I mean, we're getting really but the idea is the idea of the SWIG groups is to come up with standards of how we do the work, how we interpret it, and how people should be educated if they're going to be in this area. Okay, and so that gives some firm guidelines. You want to be a drug analyst, you have to have this kind of background. Okay, and they'll come up with SWIG drug is not actually listed on that list on the left. Oh yeah, SWIG drug down. No, SWIG dog. Okay. SWIG drug right there.
What is SWIG dog?
Okay, dog and orthogonal detector guidelines. That's basically cadaver dogs, arson dogs, with the emphasis on the arson and explosive dogs because, it's not so much, you'd like to find the remains, but, whether you're sniffing somebody to see if they might have, if there's trying to find the arson accelerant, find the explosive traces, a little bit more important. So that's what they're doing, and they develop things like, in fact, at the NIST conference I was at last week, we're talking about how to develop canine training aids. You have to be able to train the dogs to do this, and how to detect, ignitable liquids, how to detect traces of explosives. They were developing some new technologies for these training aids. SWIG drug, which is right below SWIG doc, now an analysis of seized drugs. They come up with, here's the methods of analysis we have available to us. This is the top, this is category A, these are the most powerful ones. These category B, category C, and so, you know, some rules. If you get a drug identification by category A, you have to use another method. It can be category A, B, whatever. You have to at least one other method. And then category Bs, you have to have at least 2 category Bs to make an identification. Set up some guidelines. If you're going to say you've identified this particular drug, this is how you have to do it. This all grew out of twig dam. This exists because of twig dam. In part because the attacks on DNA became the template for the defense bar to go after a number of other areas as well. And some are more vulnerable than others. Drug analysis is that's pretty cut and dried. It's basic analytical chemistry. It's pretty hard to attack. Handwriting analysis, that's a different story. Ballistic comparisons, tool mark comparison, different story. Fingerprint you would think would be on pretty sound basis. Turns out not, okay? And that's been one of the things that they have now, judges are forcing fingerprint examiners to back off a claim, say, well, I've matched the 12 points of comparison and it's his fingerprint. No, so it could be. You can't really now make that definite statement because you have no basis for claiming that 12 or 16 points of comparison tells you that it's his fingerprint and can't be anybody else's. There's no scientific basis for that statement. I shocked a judge in the first DNA case in D.C. I actually said that in court, and he sort of did a double take. I said, all these numbers that they say, so many points of comparison, they just pull them out of a dark, I didn't say it this way. They just pulled it out of a dark, remote place. It was all an administrative decision. It's not signed. There's no scientific basis for that claim. Okay. Now, there's a lot more collateral damage. DNA profiling destroyed microscopical hair comparisons. We'd use that a lot. Look at 2 hairs, look at microscopically. Now, when I did this, I would always say they could have come from the same source. I can't exclude the known person as being the source of this questioned hair. But basically, that destroyed it. The DNA analysts and the hair analysts at the FBI, they did a retrospective on their casework. And they looked at, when we had positive associations, they had like 80 positive associations, and they did DNA testing, and they found that they were wrong about 9 times. So that's 9 out of 80. That's a fairly substantial error rate. Now, there are probably some people who are very, very good at this. One of my friends, Hal Dedman, who was also one of the first FBI DNA analysts, or experts, he was also a hair and fibers expert, and he actually did a blind proficiency study where he got he got 100% correct. I mean, looking at blind samples, and he's very, very good at it, probably best person in the world, but most people are not as well trained as he is. Yes?
Just out of curiosity, do you know if they ever did DNA on the Atlanta child murderer?
No, they didn't. They had, and that could be something that he's been attempting to get that case reopened.
Because they had all kinds of fibro.
Oh, yeah. Some of them were hairs, but most of them were fibers. And in particular, the yellow trilobal carpet fibers from a carpet mill in Dalton, Georgia, as it happened. So unusual that the experts at DuPont couldn't tell the FBI people didn't recognize it, and they didn't recognize it either initially, which means it's really unusual. And the first thing they talked about was at the trial was how many yards of that was the textile industry work, how many yards of this was yellow was manufactured, how many was sent. So but they know that it wasn't just visual comparison. They did microspectrophotometry on the color and so on. My friend Hal Dedman was the expert in that case. They might get a reopened, but I suspect that might not be the outcome he would like, because assuming that they actually have some DNA that they can work from, he dumped a lot of the bodies in the Chattahoochee River. It didn't wash off all the trace evidence because they still were finding carpet fibers. As I indicated earlier, the attacks on DNA profiling became the template for attacks on the identification sciences in general: fingerprints, document examination, firearm tool mark identification. These all took a really big hit. Now, some of them really deserve to, because fingerprints, they had standards that had no scientific basis. They're still wrangling over a research project that would actually establish some guidelines to come up with criteria. How many points of comparison do you have to have? And the FBI has been able to sabotage those research grants, unfortunately.
Fingerprints. With fingerprints, did they? Don't have to. You can't really. You have to actually have a person look at them, right? You can't really.
Well, they have an AFIS system, which is automatic fingerprint identification system, and that gives you candidates, and then finally you have an analyst. But even with an analyst, how many points does he have to see? And you give with very fragmentary latent prints, it becomes.
It's like some ways DNA is a little bit easier, it's a little more clear cut than fingerprints.
It either you get the peek at the corresponding to this allele or you don't. Okay. Yes.
Along that same line, is it true that like one analyst might go by 5 points and another might go by 6? So
yeah. Although there's different in Europe from here. In Europe, in say Great Britain, it's 16 points. Here it's been 12 for an individual print. You could do fewer if you had multiple prints.
So 2 could disagree on a match?
Yeah. Well, and of course they had the fiasco, which is this is not related to DNA, but they had the fiasco of the Madrid bombing, where they had the fingerprints there that they said matched a lawyer on the West Coast, and he, I think, was arrested and so on. But it turned out it wasn't his. They got a better quality image, and it was now not a match. They decided it wasn't a match. The problem is with fingerprints, because there are various ways that, particularly if you're dealing with latent fingerprints as opposed to doing rolled inked prints where you have a really big area. The latent fingerprints are rather typically fragmentary. The quality of the print varies with the surface. It varies with the pressure. And you can always sort of squint your eyes and say, well, that's due to the difference in the difference between the inked print and the latent print is due to the way, it's an artifact. It's not a real difference. Yes? Kind of
like maybe signatures?
Yeah, it's not quite that bad, but it's still the kind of on that order. Now I'll give you an example. Collaborative Testing Service in McLean sends out proficiency test kits, and they sent out a proficiency test kit where they did something really tricky. They had some prints, and they weren't latent prints. They were actually prints in blood, so they were actually reversed from what you'd normally see. So the images were reversed. And they had fingerprint cards, 10 cards, for people that they said, try to match them up. And they had a print from one twin, and they had the 10 card of the other twin. And people were matching, a large number of the analysts matched the 10 card print to the print in blood. Identical twin prints are pretty similar. They're distinguishable, but they're very similar. And so it's real easy to kind of squint your eyes and say, well, a little difference there, a little bifurcate. That's it's just an artifact, and I can ignore it. Okay, finally, 2009, the National Research Council issued a report, Strengthening Forensic Science in the United States, where they looked at the whole thing and came up with some suggestions. Well, the thing that's interesting here from the perspective of this talk is DNA is now the gold standard. All other disciplines should look like DNA profiling. Should have the same kind of scientific rigor, documentation, training, standards, and so forth. So it became we went in from 1980s in about 30 years or so. Goes to it becomes the touchstone. It's going to be the thing that we're going to test everything else against. Okay. Now we'll see that perhaps it's a very powerful tool, but it doesn't always. It can misfire. The work is being done by human beings, and so there are all possibilities of problems. But before we get into that, let's talk a little bit about one other type of DNA. We'll talk about mitochondrial DNA. Mitochondrial DNA is DNA that's outside the nucleus. It's in these little organelles that actually provide energy for cells. And there's a loop of DNA here, and that's what it looks like. It's about 1,600 and something base pairs. Contains a lot of has genes on it that are related to energy production in cells. And up there where it says the D loop, there's a hypervariable region up there that doesn't actually code for anything, and therefore the mutation rates are several hundred times higher than everywhere else. And so people started to look at this. Why is this interesting? Well, because it's multi-copy. You have hundreds to thousands of mitochondria per cell, whereas you have one nucleus, and the nuclear DNA is single copy only. So if you get to degraded samples, maybe the nuclear DNA may not be salvageable, the mitochondrial may very well be. So we do sequencing on it. I won't go through it. It's basically we just use PCR. We put modified sugars in, bases with some modified sugars in them. These are labeled. They're modified so that the chain doesn't get extended past them. They get incorporated at random, so you wind up with a collection of different sized segments with different terminators on them, and you run them, do gel electrophoresis on them, and just read off. You just read off the gene sequence. So it's really kind of cute. Yeah, all of this is really kind of fascinatingly clever, science. Okay, a couple of facts about it. Mitochondrial DNA is maternally inherited. Comes from the egg. The maternal DNA may get so diluted, apparently if maternal DNA gets into, mitochondrial DNA gets into the cell, it gets labeled so that it's attacked and destroyed. So it's all coming from the mother, which has one unfortunate feature, and that is, it means that any pattern, any sequence you see in a particular individual, it's not unique, because his mother had it, and all the maternal line all the way back, and all siblings will have it too. Okay, it's multi-copy, but it can be used on samples that don't have a lot of DNA for various reasons. Bone, dealing with skeletal remains, get the mitochondrial DNA out of the bone. You can do teeth. When my colleague Jim Starrs dug up Jesse James, or what was supposed to be Jesse James' body, they tried doing mitochondrial DNA out of the bone. They couldn't get the same sequence more than each sequence. Every time they got the sequence, it was different. They cracked open the teeth and took the dental pulp out, and they got a nice profile, matched James' family members. You can do hair. Yeah, it was Jesse James. Yeah, all the evidence points to him being Jesse James. The bullet in the 36 caliber pistol bullet that was still in his chest. This was back. He got that in 1865. He encountered some U.S. Army troops, and he was an unlawful combatant. So they took the simple measure. They just shot him, and they didn't kill him. His commander, William Clark Quantrell, encountered federal troops. They killed him. Well, you deal with unlawful combatants, you just kill them and be done with it. Anyway, the fact the mitochondrial DNA typing of hair is one of the things that killed the microscopical comparison. Now, give you some examples of how the mitochondrial DNA might be used. Take a case for local again. In 1994, President Reagan honored this supposedly unknown American service member from the Vietnam War. He was interred as one of the unknowns in Arlington National Cemetery. 10 years later, a POW MIA activist claimed that the remains were actually those of First Lieutenant Michael Joseph Blassie. And there was they had actually supposedly did a skeletal identification at the Central Identification Laboratory in Hawaii, what's known as SILHI. And there was a scandal that blew up. Apparently, the anthropology work there was really crap and was really bad. And this was done like 6 bone fragments, and they made this identification, try to destroy all the evidence, which is what they normally do anyways. They destroy all the background evidence on the unknowns. But there was this activist was able to find enough information to suggest, well, there's a part of a flight suit and some other stuff, and he suggested from where it was located and the other facts suggest it was Blassie's remains. Then they did mitochondrial DNA profiling, sequencing, and confirmed were those of Blassie. That's Blassie there. He was shot down near An Loc in 1972. And probably because of the fact that the active duty members of the armed forces have to have their DNA on file in a register, a repository, there won't be any more unknown soldiers, because everybody whose any remains are recovered will be identifiable. And the only way you'll have people missing in action who are just basically vaporized and there's no trace of them, but they're not. You have to have some remains to actually bury in Arlington. Okay, some other things new things that are happening. We've got Y chromosome SDRs. Big thing right now is touch DNA. We had a speaker from the Bureau of Alcohol, Tobacco, and Firearms. They've set up a DNA unit. They look at where people touch things. You handle stuff. You guys have been, putting your hands on the arms of these chairs. You've left enough DNA to get a profile off of that. Thank you very much. And I'll be going around and swabbing everything. And where it's interesting is where people you can get it off of incendiary devices. A guy will make a Molotov cocktail, but he buy a wine bottle. He'll drink the wine, leave DNA on the lip, throw it. It doesn't break up, or you recover that part of the bottle. You can just swab it. He constructs an IED, and he leaves his DNA behind. The military's been doing this for years in Iraq, and the ATF does that too. So you do the Ted Kaczynski thing, sand everything off before you place the bomb. Okay, looking at microbial DNA, chemical warfare agents. Human microbiome, you've got all sorts of little microorganisms that you can leave behind. This is almost as good a marker as your DNA. And some of these markers you can look for some different microfauna that are present, like in vaginal fluid, and you can identify some of these body fluids that way. In case of vaginal fluid, we've never had a really good method of identifying it in a stain. And then we are looking at these SNPs, these single nucleotide polymorphisms, where there's just a single base difference. And two of the types, we have a researcher who's looking at phenotype informative SNPs. I'm one of the subjects. These tell you hair color, eye color, skin color. That now gets into some privacy issues, but it can be useful. You can make some inferences about the appearance of the unknown source of DNA. He's going to look at some of the SNPs associated with the genes that govern craniofacial structure. So you may be able to combine the information to produce a likeness of the person. Yes?
I'm sorry, could you speak a little bit to the reliability of the microbiome data? I mean, certainly some people only differ in terms of quantity of different...
Well, that's what they're looking at. That's why it's a research area. Nobody's tried to use this yet, but we had a researcher from Italy gave a talk at the university recently, and they have to do all of that kind of background data, show that it made quantity. They're doing some things where you can do quantitation with like real-time PCR, the things like that.
That's a compelling idea.
Yeah,
it seems too soon
yeah. They're not doing it yet in casework. The ancestry informative SNPs, you can also say something about the biological background of a person. And the one that really gets into some interesting aspects of privacy issues about DNA labs on a chip. The NIJ has actually funded several prototypes. We basically go to a crime scene, put some blood or sample there, and it actually churns away, does a DNA sample, does a profile, sends it. You have an uplink to, internet, send it back to the lab. You could also do suspects, people that you've just apprehended. Just some way you could take their fingerprints, you could take a blood DNA sample, and submit it to CODIS. That's going to be very interesting to see what happens there. Okay, you've heard probably about the Y chromosome STRs. They're paternally inherited. They're inherited as a block because you only have one. A male only has one Y chromosome, and supposedly showed that Jefferson, fathered one of Sally Hemings' sons. A very compelling argument has been made this is a misinterpretation. Some member of his family did, probably a nephew, is much more likely in terms of access and being around Hemings and having access to her, which Jefferson would have been much more limited than this person. Another cold case. This one is still cold, but it's kind of interesting because it illustrates something about databases that might be online. This is a 1991 murder. She was murdered on a high school campus in Seattle, Washington. In 2010, the perpetrator DNA was profiled, and then the information was submitted to an online genealogical database where you can post DNA profiles, and it matched to the link to the family of Robert Fuller, who arrived in Massachusetts in 1630. Now, this isn't terribly helpful because there probably a genealogist estimated there were probably looks like 34,000 male descendants of Fuller. In some places, it might be even worse. There's some places where, in the areas that constituted the old Mongol Empire, it's estimated that 8% of the males have the STR profile of Genghis Khan. Yeah. Okay. Okay. And we are almost done. I'm going to get to this one right here. This is the final case. I'm going to make. This is the Mixer case. She was murdered in 1969. She was strangled and shot to death. Her murder was attributed to the Michigan co-ed murderer, John Norman Collins, who was convicted in 1970 of one of these crimes, but not of the other. So it remained a cold case. So they did 2002, the state of Michigan did they conducted DNA tests on her pantyhose and a spot of blood from her left hand. Mixer's DNA was not detected on her pantyhose. The DNA on the pantyhose was believed to be either saliva, sweat, or skin cells. It was matched to a man named Gary Leiterman. He was in the database because he had been kiting drug prescriptions. Okay, he's arrested, convicted of the murder. Okay, so good. It's a DNA triumph. Okay.
Is that the picture of him?
Yeah, that was him with his grandson. There is, in fact, an organization there that believes he's been wrongly convicted. Why? The spot of blood on the hand, the DNA there matched convicted killer John Rolas. Okay, that maybe they're working together. Except that Rolas was 4 years old in 1969 and lived 40 miles from the murder scene. Okay. Now the prosecutor still maintains he was really involved. Okay. But the problem is that the evidence from Rulis's case was processed in the DNA lab about the same time as the Mixer case. So, we have possibilities. What could go wrong? Mislabeling of samples, cross-contamination of samples, fabrication of evidence. In one of the cases where Fred Zane was involved, there was a DNA match. This was actually in the not the Oscar Davis case. Well, there was one there, but there was another case I discussed with people from Cellmark. And they said they were convinced that they put the known sample in the lane in the known lane, and they put the another score to the known sample in the question lane. So they got a perfect match, but it was the result was totally fabricated. So at this point, I think we are ready to go to have.
Let's do some cute questions.
Okay, we take some questions. Okay, yeah.
What do you think in terms of the standards that needs to be applied in terms of privacy? You mentioned that a few times. Do you think there needs to be any standards for the presumption of innocence and due process, or do you think that that's more of the obfuscation defense lawyer?
Well, I think I'd restrict it to people in the database, to people actually convicted of something. I think that there's a good bit of case law. Or there's not a good bit of case laws, but there's case law that says if you put somebody in a database, like in the Fingerprint database, this happened to a man named Gary Rank in Pennsylvania, he successfully sued to force the FBI to take his TIN card out of the database because he was exonerated in the case in which he was arrested. So why do we, open ourselves up to having to go and erase data out of the, take it out of the CODIS system? And if the person's not guilty of anything, why would he want him to be in the database? So certainly one that's going to use for criminal investigation. Yes. Yeah.
I was wondering if you could say anything about the SAFER Act that's possibly that's in the process of being voted on now. If you know anything about that. It's, about the untested 400,000 possibly rape kits across the country.
Well, that's the thing. They don't want those thrown away. They want to go back and try to process them, but it's going to take a lot of money, a lot of time. It could well be done. In fact, the last year of funding under the 1994 DNA Identification Act, we actually, the solicitation said, tell us how you're going to get down your warehouse databases, your warehouse samples. Tell us how you're going to do that. And then we were granting money for people who were going to try to attack this. People don't know that, but because they were unknown subject cases, the most that was done was to see if there was evidence of sexual activity. There's no point doing anything else until CODIS existed. Now this stuff is all just sitting there, just database. They need to go back. It would be helpful to go back and get it, take it, and see if they have some people that need to be in the CODIS system that aren't. Yeah.
Have criminals, when did they start, or did they start to compensate for these things?
Well, it took them a while to catch on, then they started to do things like use condoms. Of course, then what they would do is leave the condoms at the scene. That's what happened. They had multiple rape involving a prostitute who was abducted from the District of Columbia, brought to Northern Virginia, and they raped her using a condom, and they left the condom behind, which is like the pristine, DNA evidence. Thank you very much. Brilliant. They try to do things like wash up, use bleach, try to destroy the DNA if they possibly can. The problem is, you got to cover everything. If you didn't wrap yourself in latex and like a big condom and not touch anything, you've probably left something there. And so there's a limit to what you can do.
So has this increased real convictions, or what is this? Has it ever done any sociological?
Well, it probably doesn't increase the number of. One of the things it probably does is probably permanently remove people who committed crimes. The FBI did a study back in the beginning of the Reagan administration. They realized that 75% of all violent crime was committed by 25% of the criminals. So if you bag those people up and use the 3 strikes and they're outlaw, just put them in jail and kept them, well, crime would have to go down because most of the bad guys are rotting in jail, in prison. Maybe not. I don't know if I would go that far. And there's always new people entering the field. The young males that come in, that decide they're going to take up, rape. Well, they're going to take up rape as a hobby, sort of thing, or an avocation. Yeah.
You mentioned a couple of times evidence of sexual activity. What does that mean?
Basically means semen on a vaginal swab. Okay. Now that could be consensual, could be a spouse or significant other, could be, it doesn't mean that there's evidence of a crime. You don't know until you get further information, which can often be a problem because, in the case of what was Crystal Mangan in the Duke lacrosse case, she had DNA of 3 different males in her. None of them the members of the Duke lacrosse team. Yeah.
You mentioned the Ancestry database. Things like 23andMe or advertising, find out your ancestry, see if you're descended from Genghis Khan. What do you think of the use of DNA for health profiling individually and for ancestry? Obviously not forensic.
For profiling, it's probably going within probably a decade or so. It'll probably be considered bad, medical practice if you don't do do at least some DNA profiling, because people, to see if they, process drugs differently. That's going to be. I've had discussions with several of my colleagues, and they say that's, it will be malpractice if you don't do it. Yeah, individualized treatment. Really, that would be very helpful.
The ones now promising your ancestral profile, do you see validity there?
Well, it depends. You may actually the only way you really know about your ancestral profile is to there are legitimate databases that you can look at, but you have to do stuff beyond what they're doing. But you can do that. There are some actual ones that are run by the government, and you actually know how they got in there. I mean, you don't know about some of these others, who submitted them, how accurate their background information is. I don't know about the identifying the Fuller family. Somebody put that in, so they were descended, from somebody. They got some sort of family tree. But in the case of STRs, YSTRs, there was a study done in England where they looked at families living in a particular village, and they looked at the STR lineages, and they found what they primly refer to as a number of non-paternity events. And you can sort of figure out what we're talking about. That was the that's the case. The person may not be who they think they are. There are all sorts of little, family secrets. It can be valuable, but it may be very misleading. More in terms of not specific lineages, but perhaps, areas where, Western Europe, whatever, that sort of thing. Might be more interesting. Yeah.
Time for one more. Could 23andMe be, that database be used for identification purposes, if you get a forensic way?
Well, you saw what happened. They can narrow it down to one lineage, okay?
The database
They got 34,000. You could narrow it down, but it doesn't tell you very much, okay? What the CODIS tells you, it says you get a match, you know who that belongs to. If you get a close match, and we have to come up with some criteria. What's a match that you would consider it likely it's a family member? You've got a family name. You've got somebody there that you can go and look at. On these other databases, you got whoever submitted. That's probably the most you've got. That person may have nothing whatsoever to do with the case. Okay, and certainly would resent your suggestion that he did or she did. So that was an interesting use, and it makes a point. I don't think I would want to put anything in one of the any genetic information in there. I think I'd be really leery about that. Unfortunately, I can't control that because other members of my family might do something like that. So you have limited control over it. But the CODIS system, because it identifies the DNA to specific people. It's actually two parts. There's one that's the offender database, and that identifies the specific people. And then there's also open case, DNA that hasn't been matched to anybody.
Thank you very much, Walter.
Thank you.
We have